I would like to thank to Human Frontiers not only for collaborative spirits, but for being catalytics to initiate this project. It's really unanswered questions, and it has two components. Well, in fact, how from all of this we can get this? In fact, it is mind-brain on this picture. And more importantly, I try to address two questions. Why and how did cells come together to work together? So there is neurobiology of animal origins. It's inseparable from origin of neurons itself. And at the same time, if Earth's history were to repeat, could animal evolve again? Would neurons and intelligence emerge? And if so, in what type of forms? So this is the fundamental questions about evolution of an integrative system. These slides represent major eukaryotic lineages, and most of them unicellular. So multicellularity is the disadvantages. It's only 5 lineages were able to achieve complex multicellularity, and these are green, red, and brown algae, fungi, and metazoa animals, which become extremely successful in terms of speciations between 10 and 100 million species. So you can ask questions, why is that? And narrative and framework lies in feeding revolution with new integrative systems able to deliver 500 signal molecules per species at the very minimum, and up to 2,000 and 3,000 signal molecules at average in bilaterians. So I'll try to follow advice from organizers and Human Frontiers. Do not go to particular technical details, and try to have a broad overview, which will be probably important for many of us. So recent analysis from nearly 20 phyla came to these conclusions. Neurons in synapses evolved more than 3 times. Complex brains evolved more than 20 times. Muscles 3 times. Guts, gastrulations, formation of the germ layers, other recognitions. And in most of this is the logo toolkits, a variety of the signal molecules which functionally glue cells together. And as a result, there is enormous convergent evolution, but consensus goes that all animals, all of the 34 phyla, evolved in relatively short intervals, approximately less than 3 million years, because 3.5 billion years required to accumulate toolkits, mostly because of plate tectonics. So we try to reconstruct the origin of this integrative system, but most of the interesting animals we cannot simply bring to the lab. They are fragile, or they don't want to live in the lab. So we came with a concept which called the shipseek, but right now it's developed into a physiological aspect. We actually made laboratory on the boat and use Starlink to do satellites connections to supercomputers, so you can get fresh animals for virtually different time of analysis, including behavioral development, genomic analysis, and at the same time, in real time, interact with supercomputers. And this is still ongoing efforts. Here, what we try to do conceptually different, rather just to do simply knockout, which everybody uses, but there is a redundancy of signaling pathway, we try to take advantage for experiments already performed by nature, which work on the systemic level, and 99% of bad experiment was eliminated. And secondly, equally important, we're able to do experiment in real life, in real habitats, which surprisingly very different from standard luxury conditions which we have for animals in the lab. So this is sort of like a trace in Darwin with a type of Star Trek operations. And we also just end up with concept of labs, which can be for 5,000 rows delivered to any other place on the planet, or also operate on the boat. And key aspect, what happens during the last year, talking about methodology, we work very tight with chemists and develop protocols which are massive single-cell multi-omics. I called it 4D+. And the reason for this 4D+, because it's spatial, time and evolution. So what is possible to achieve today, in parallel for 150,000 cells, for every cells, do chemical analysis for up to 1,000 proteins, metabolites, close to 1,000 lipids, and at the same time do transcriptome. So in this case, you heavily rely on AI to analyze this complex data set. And we have successes with several species, and I will illustrate some summaries. So in addition to classical theory, which is the textbook about the region of animals, which called like gastrea, so currently there is a different perceptions. Cell types predate multicellularity. So it's not vice versa. When you have simpler organism and then you have multiple cell type. It's completely reverse. So there is a complex life cycle which have temporally separated different type of the stages, and there is a several species carefully analyzed for that. And this temporal differentiations was transformed to spatial differentiations. So toolkits is already there. You only have to do collections, and this is a major basis for convergent evolutions. And another insight, when you can do single cell multi-omic, it's every cell is secretory cell. So every cell releases bunch of the signal molecules. And I will illustrate it with this metabolomic analysis, which is only tip of the iceberg. So when you look at this actual concentration of metabolites in every cell, look at this glutamate, nearly like 60 up to 100 millimoles. So in fact, in our cells, it's not potassium chloride, when you can do it in the textbooks, it's a potassium glutarate, and usually most of the 10 different kind of metabolites or amino signal molecules, and ATP is real signal molecules. And when we try to combine data together, this was shocking. There is about 20 million signal molecules existing across eukaryotic kingdoms. And at least for animals, we have order of 100,000 signal molecules. So these put everybody in the different perspective, not rather to look in the one or another genes. It's basically how all of this complexity work together. We do have alphabet, but we don't have idea about syntax. And to resolve this sequence, we clearly have to go to relatively simpler animals. This is recent reconstructions of evolution of neurosystem. Oh, sorry, not animals, new phylogeny. And this was shocking because this lineage of animals, which called ctenophores, come jellies. They have complexity comparable to bilaterians, and they apparently branch earlier. And then they have neurons, muscles, synapses, mesoderms, gastrulation, etc. Then we have simplified animals, and finally we have like another design represented by my dog, Betsy. So in fact, this conclusion is apparently solid. Neurons evolved more than once from secretory cells. And I even go further. It looked like that from our analysis. I would like to discuss details if you like later. You cannot go to these detailed slides. So apparently, signal molecules not only made the neurosystem, not vice versa. Signal molecules really made animals. That is neurobiology of animal regions. And obviously behavioral, which is controlled by a variety of the signal molecules. It's a pacemaker of evolutions. So next step, this slides, in fact, summarize nearly like 6 years of research. It's rare relationships between different kind of neurosystems. So 2 points. This side indicate independent region of neurons. And these stars indicate independent regions of the complex brain. So we documented 23 ways how you do centralization to make a complex brain, and the unique combinations or syntax of signaling systems. So currently, major role in this plays neuropeptides, which is the small secretory molecules in which in our brain and brain practically any other organism on the order of about 1,000 working all together with a complex gradient. I will not be able to go to any individual lineages. Understandably, there is a very short talk. I will just only show like an example from some most interesting example, like early branched complex animals, ctenophores, using both and some another one. First time for the 100 years, we basically get inventory of all described species and their phylogeny, which is put over here. But what is interesting about these animals, this all life is made on the cilia. neurons involved to control cilia. This is a mouse of this ctenophores. This is elementary brain. This is a conductive pathway. So I just really call them aliens of the sea because the way how they operate all together. But even more surprisingly, these guys have not one, but two neural systems which independently evolved. One of them in the skin, and second is the mesoglia, which sometimes look like immune cells or muscle cells with multiple trans differentiations and separate regions and separate toolkits, and none of them overlap with the rest of the animals. So considering phylogeny and unique molecular blueprints, including master regulator and signal molecules, it's a solid case that this neural system evolved independently. So we try to decipher it with multi-omics. Of course, it's impossible to go to all technical details. So it's about 500,000 cells analyzed. And we pick up altogether 5 different lineages of neurons, which genealogically came together from different types of the sources. And again, for each of these lineages, no similarity to the rest of them. So they're true aliens of the sea, which independently developed neural system and convergent neural system. But even more interesting, these organisms have reduced immune system and reduce our recognitions. So what it was found, reported also earlier, we can make a chimeric neural system. We can link and fuse animals because it's low our recognitions within hours, sometimes up to one day. And very recently, basically a few months ago, we showed clear evidence that neural systems fuse together and work together and communicate and establish completely new hierarchy. Even more, some fragment of these animals form a neuro-bot or a ctenophore, small living machines from about 1,000 cells, 300 cells, 100 cells, which can learn, remember, and navigate. So it's a really minimalistic case. And final surprise, also this year, a couple of months ago, we were following this and low immune system, we were able to make chimeric neural systems and chimeric animals from different species. So we currently have evidence that for 6 different species of ctenophores, most amazingly that within 1 or 2 days, somehow, even they belong to different orders and families, they establish communications and work together. Our extreme examples, which still live in the aquarium, it is 5 animals fused together and the neural system working together. So it's an ongoing process. And this is my last slide, because this is alternative neural system. There is another example of animals which called the placozoans. It's only about 6 morphological cell types. And these organisms have more than 30 different types of behaviors, including complex behaviors. And when we did this kind of molecular single cell multi-omic analysis, we ended up with more than 40 cell types. And some of them look like pyramidal cell types, and they make like 2 different kind of meshworks for complex and coordinated behaviors, which is unrelated to anything we'll now again. So I have a couple of another examples, but considering to the time, it looks like evolution have more or less 5 designs of integrative systems. And this is the best experiments. It's like working on real aliens in time. What is most surprising in this case, and I think this is my final slide. Most surprising in this case that even these minimalistic animals has 183 secretory molecules. They have special cavities, and they create chemical gradient, which in fact, without synapses, established hierarchy of the behaviors and its plasticity. So I think these are real prototypes how signaling evolved, not electricity. It's a real chemical brain based on the gradient. And at this moment, we know that at least 100, 200 molecules always work together, even in this pre-animal stage. And with that, I would like to thanks funding agency and again, Human Frontiers, which initiated this project in an unpredictable way. Thank you.